Assalamu alaikum. Good morning. Okay. Good morning, everybody. We'll continue with this same experiment that we started before. Now, Nassar, can you please read the title of today's experiment, of today's lesson? Wait. Gene cloning. Now, in this title, there are two keywords, which are, what are these two keywords? Yes. And what is the second word? Gene. Now, what does a cloning mean? Yes? Yes, making millions and millions of copies. What thing we are going to clone in this experiment? Shahawani? Gene. Can you name exactly the gene that we are going to clone in this experiment? The gene for Canomicine, the Canomicine gene. What is Canomicine, Tariq? Do you know Canomicine? Nassar? Is it something we eat? Well, Canomicine, this is the same as ampicillin, tryptomycin. Do these names tell you something, remind you about something? Yes. Antibiotic. The Canomicine is what? Is antibiotic, ya Nassar. Now, this is the overall experiment we are going to do. Now, it is divided into modules. Today's module is about what, Rashid? Transformation in E. coli. Can you remind us what does transformation mean? What is transformation?
Transformation when you take one part of DNA.
Okay. Okay, that's absolutely true. Transformation, this is when we force a bacterial cell to take in extragenous DNA from outside. This is transformation, okay? Now, and in today's lab we have two main objectives. Muhammad, can you please read the first objective? In vitro. Yes. Extraction and restriction enzyme analysis. Okay, these are the overall objectives of the experiment. We are going to do a recombinant DNA plasmid. We are going first to do a recombinant DNA plasmid, and this is we did last week. This is we did a week ago. The recombinant DNA plasmid. Now, what does recombinant mean? Recombinant DNA. Recombinant. What does recombinant mean? Yes, Imadi.
You take DNA
from two different sources, and the DNA that we recombined in this experiment a week ago was what were the two different sources of DNA that we used? We joined what and what? Yes, Tamimi. We joined what and what? With gene. What kind of a plasmid we used? PUC plasmid. And the gene, the Canomicine gene. We linked them together. How we linked them together? Using what? Yes, Nasser, Shahabik. What did we use to link the ligase enzyme? The ligase enzyme. Ligase enzymes joined or make bonds between the separated pieces of or fragments of DNA. Join them together. Then, how can we analyze, how can we know if the recombination was successful or not? Gel electrophoresis. Did we do gel electrophoresis, Tariq? Nassar? Yes, we did. Now, I will show you the results of the electrophoresis. Okay, then we will discuss it together. This is the gel that we did. that you did last time. Okay. It is much clearer in the gel itself than it is here. Now, look at these lanes, at these different lanes here. In this lane, in the first lane, how many bands can you see? One. In the second lane, in the third, in the fourth, two lanes you can see, right? The last one, this is the ladder. The last one, this is the ladder. Now, why do we have here only one lane while here we have two lanes? Yes, Shahwani. Okay, so you want to tell us that this one lane represents what? It is what, Imadi? This one lane is what? Is it the two pieces of DNA joined together or separated from each other? They are. When they are separated, you expect to have the PAC and the gene, the Canomicine gene, to form one band or two bands. Two bands. So here in this lane, this band and this band would stand for what and what? For the plasmid and the Canomicine gene. While this band or this band or this band stands for what? Amadi, yes? For these two things, the plasmid and the DNA, but joined together, linked together. When they are linked together, we describe the plasmid as what? Recombinant plasmid. Recombinant plasmid. Now, why these bands migrated less, moved less distance from the well than these two separated bands? What do you think, Marie? Why this band moved a little distance compared to this band? This is the well, this is our reference, okay? This band moved only this distance, while this band moved a bigger distance. Is this related to the size? What do you think? When two individuals are like making a race, who would move faster? The thin or the fat one? The thin one will move faster. So this is the distance that they migrated is related to what? To the size. Which one do you expect to be bigger? This one or this one? The first one. This one is bigger. It migrated less. It is the plasmid and the gene joined together. Two things together. Here they are separated. Here they are independent. Okay? Here they are not linked together. But here when they are linked together, they formed one big piece. That's why it moved a little. Okay? Now, in today's experiment, what we will do? We will, this recombinant DNA, we will what? We will transform it into the bacteria. We will force the E. coli bacteria to take it inside. Okay? before doing that, I would like to invite you to come in pairs, okay, and to see the gel here in the illuminator. Okay, come Nassar and Rashid, have a look at the gel, at the bands. Now, can you tell us what does these two bands mean? This one and this one. What do these two bands mean? Represent what? They represent what? Separated DNA fragments or recombinant DNA plasmid? These two fragments, they are separated. They are separated. That's why we have two things. Here you have the recombinant. Ahsant Nassar. That's absolutely true. Now, these are the wells. Okay? So, the recombinant one migrated or moved less distance. Why is that? Because their size is more or less? Is more. When they are joined together, they are more. They are bigger, so they move less. Okay, step back, please. Now, can another two come, please? Have a look at the bands. Have a look at the bands at the illuminator. Okay. Again. Can you tell me what do these two bands represent? Yes, they are represent the plasmid, the Bach plasmid, and the gene. What is the name of the gene? Canomicine gene. Now, Muhammad, what do these bands represent? It is DNA. It is all DNA. Now, recombinant or separated? Recombinant DNA plasmid. They were ligated together. Now, why do the recombinant plasmid migrated less distance? Because they are heavier. Okay, thank you. Step back, please. Now, the last two, come, Nasser. I want you to see the bands now. Okay, let's examine this lane. Now. What do these two bands represent? DNA, okay, DNA fragments, DNA pieces, right? They represent the ligase enzyme and the PAC19, okay? Now, here they are separated. These bands represent the recombinant DNA plasmid. Which is heavier, which is more, has more molecular weight? The recombinant DNA. That's why it migrated less or more from the wells? Less distance, okay? Later on, by the end of the next lab, you will measure these distances and you will plot the graph, okay? Thank you. Step back, please. Step back, please. Now, we will resume with the work in the lab. To do so, we will review quickly the procedure. So again, after we have visualized the results of the electrophoresis, now I will invite you quickly to watch a short video about the transformation process to recall the procedure, how it is implemented. Then we will start with the process itself, okay?
Of creating human insulin for diabetic patients. To this point, our efforts have been focused on creating a recombinant DNA plasmid that will be able to express human insulin. But, if you recall, our ultimate plan is to get bacterial cells to make insulin. That means we need to get our human insulin plasmid into a bacterial cell. Is that even feasible? Well,
Now, how is this step applied to our experiment? What kind of a plasmid we are using? Name the plasmid. Yes, please. Yes, PUC, the puck. Now, and what gene we are going to clone? The gene that we are going to clone, yes? The Canomicine gene, exactly.
Lucky for us, there are a number of ways to genetically modify bacterial cells. One of the most common procedures used in laboratories and classrooms alike is known as transformation. Transformation is the process in which the genetic makeup of a cell is changed by the introduction of DNA from the surrounding environment. Scientists commonly use the bacteria found in our gut in bacterial transformation experiments. This bacteria is known as Escherichia coli, or E. coli for short. E. coli is one of the most commonly used organisms in scientific experiments. Bacterial cells that are actively growing are most amenable to bacterial transformation. However, they must undergo a series of treatments before they are competent for transformation.
Now again, to convert the bacteria, to convert the E. coli into a competent bacteria, what things we need to do? Who can remind us? Yes, Shahwani. Calcium chloride. What do you think, Mary? Why we need to add calcium chloride? To break down the cell membrane. Now, Ahmadi, do you agree with him? No, not exactly. Not exactly. What you have said is true, but it is for another treatment. We need to create gaps in the cell membrane and the cell wall, but not using CaCl2. The calcium chloride is used for what? To neutralize the charges. The charges of what? The cell membrane and the DNA, because both DNA and cell membrane are negative charged, and negative charge, they do what? They repulse. They do repulsion, right? So to overcome this problem, the problem of repulsion, we neutralize the charges of DNA and the cell membrane using what, ya Marri? CaCl2. Absolutely true. Now, we will start with the procedure itself. So far we started preparing the DLR tubes, right? We prepared the first DLR. Who was responsible for this step? Who made the labels? The DLR, Tamimi. Yes, can you please join me here in this station? But keep distances, okay? Keep distances. So far, we have labeled the DLR tubes, dilution, ligation, reactions. Now we will start adding the components. We need 5 microliters of the DNA. We did this. Did we do this step? Nassar, do you remember? Did we add 5 microliters?
We added 5.
Yes. To which one we added? Show me, please, because you did this step, right? To this one. Okay. We added to this one. Good. Now, we need to add another 5 microliter to prepare another DLR. Okay. Come, Muhammad Tamimi. Take 5 microliters. Take 5 microliters. 5 microliters of the DNA. Of this DNA. What tip you are going to use?
5 microliter tips.
With the white tips, right? Now, take 5 microliter, add them to this tube. Help him open the centrifuge tube. Yes, to the first one. Add to the first one. Mr. Bاسم. Now, the next thing, we are going to dilute these 5 microliters with 45 microliters of water, of ultra pure water. Note, I want you to learn how to read the procedure, how to follow the procedure. Okay, you need not to memorize the procedure. You just read it through and follow it up. I know this is a long procedure, but you have to be a little more organized. Now, I want another student to take 45 microliters of distilled water and to add to the DLR tubes. Okay, yeah. 45 microliter. Which micropipette you are going to use? This one. Why this one? What is the maximum? Okay, this is 50. Now make it 45. Okay. Take a yellow tip. Now, open the distilled water. Take 45 and add them to the tubes where you just added the add here and here. Okay, good. Okay, now discard the tip. Take another tip. Add another 45 microliters to the other centrifuge tube. Okay, very good. Discard the tip. Good. Now, I want you to add more 45 microliters of highly pure water to these green capped centrifuge tubes. I want another student to do this step. Who would like to participate? يلا سعود نصار. يلا. Come. So this is the distilled water. Take 45 microliters and add them to this. Help him open these centrifuge tubes. كلهم. All of them. distilled water. بيكر صغير.
اتوقع الكمية صغيرة.
45 microliters. هذا كمان distilled water. لا don't pour here. Use directly from there. سكر هذا. التو تيوبس هذا حطيته فيهم سنترفيوج.
باقي لا ما حطيت. انا حطيت كده.
Good. Good. Put it here. Now, the next step, we will add transfer 500 microliters ice cold CaCl2. So, where is the CaCl2? Come, Imadi. Your turn. Yeah. Take the CaCl2, the calcium chloride, and look at the procedure. What is the volume you are going to add? 500 microliters. So, which pipette is more suitable? Okay, good. Now you will be using the blue tips. Okay, take 500 microliters of the calcium chloride and add to the green ones. Open them. Open the green bottles. Shahwani, can you remind us why we are adding the calcium chloride right now?
We are adding it to neutralize the plasmid and the thermometer.
Yes, to neutralize. If we don't neutralize these two things, what would happen, Rashid? Yes. Okay. على الجرين. على مين ضفت؟
ما حط بدخل تهيئ
ايوه مش على هذا، على الجرينز. To the greens. Now, back again. If we skip this step, what do you expect to happen, Mary? Repulsion. Yes, repulsion between the DNA and the cell membrane. Good.
Yes. Why we are not working under sterilized conditions?
Because we're still not using the bacteria. Now, the ready tubes, I want you to place them on ice. On ice here. Place the ready tubes on ice. The ready tubes اللي أضفت لهم الجرين tubes these. Now place them on ice. Okay. Also there are two other centrifuge tubes here. Add to them please the calcium chloride. Now the next step, who was asking about the sterile conditions? You? Now go and prepare the sterile conditions there. We will continue working on that bench, the next step. And clear it please from any papers, clear the bench there. No papers, no pencil case, nothing there should be. Now, Mary, help him with the sterile conditions there. No problem, no problem. It's calcium chloride, nothing there. Yeah, خلاص, okay. Now, place them on ice. Okay. Now, take the ice bucket there to that bench. We'll continue working at that station now. Shahwani, can you please bring the source plates, the bacterial source plates and the Canomicine plates to this area, to this bench? خذ مدفتين, take them twice يعني. Imadi, now the other Canomicine plates, please bring them there closer. Yeah, حطهم على البنش. Place them on the bench. Put them there aside. Now, let's examine these plates. Mr. Bاسم, can you please the visualizer? Turn on the visualizer there. Now, before you came to the lab 2 days ago, me and Mr. Bاسم worked on this, prepared this plate. These white dots, what do they represent? What do you think? Growth of what? Growth of bacteria. Growth of bacteria, of E. coli. Now, you are going to do what with these bacterial cells? The term, what is the term? We will add the recombinant plasmid, the gene, okay. What do we call this process? Transformation. We will transform them with what? With the recombinant plasmid. Type. Now, I also took some bacteria and plated them on this one, on this plate. Now, this plate is can you observe any growth in this plate? Now, can you observe any growth in this plate? No. No growth in this plate compared to this one. Okay. Now, what do you think? Why the smaller plate showed no growth? What might be the difference? The critical thing? More fingers. You know. Don't be shy. Answer, even if you make a mistake. Mary, I like to hear from you. What do you think? One plate showed growth, the other one did not show growth. Why is that?
There are some bacteria in the large one, while the small one didn't have.
So, you think the problem in the bacteria or in the media? In the bacteria. What about the media? Could be there something in the media that... Yes?
Human error.
Human error? Oh. Okay, Shahwani. Okay. The small plates, these are Canomicine plates. These are Canomicine plates. Canomicine plates. Canomicine is what? Where is Nassar? Where is Nassar? طيب, okay. Then Muhammad Rashid, what is Canomicine? It is antibiotic. It does what to the bacteria? Is it good for the bacteria? It kills the bacteria. That's why there is no growth here. Now, what is the term given to this plate? From an experimental point of view, when we design an experiment, we need to run what? In every experiment, there must be, besides the experimental thing, the control. So this is a control, the control. Now, if there were some growth here in this bacteria, then I would conclude what? The antibiotic is not working well. The Canomicine is destroyed, is broken, right? But the Canomicine, I can confirm that my Canomicine is very good. Why? Because there is no growth. So this was not a human error. On the contrary, this was a very good result. Okay. Now we will resume with this thing. Okay. We will transform the bacteria. We'll start transforming the bacteria. Okay. We need to check the procedure again. Now, bring the toothpicks and the loops for the transformation. Okay. Now we will do what? Now, a transfer 500 microliters of this is we did. A place tubes on ice. This is we did. Now, using a sterile inculating loop. One of you, show me the sterile inculating loop. Show me. Yes, these are the sterile inculating loops. You will use them to to transfer 5 well isolated colonies. You will choose 5 well isolated colonies from the source plate and transfer them to the test tubes we have just prepared with the calcium chloride and with the recombinant plasmid. Okay, and then twist them. Okay. Now, do you want me to demonstrate this process for you, or you can do it? What do you think, Muhammad Khalid Tamimi? You will choose 5. Bring this. You will choose 5 well separated colonies. Okay. Good. And transfer them using the inculating loop. Okay. To the tubes we have just prepared. Okay. With the green caps, and there are 2 others. This one. It's here. Okay. Now, في toothpicks. There were toothpicks. أحسن. Toothpicks there. Okay, no? Sterile toothpicks. نقلتهم. Okay. جيب. We will use the toothpicks. No. Which of these source plates you are going to use? Decide. Which one do you like more? Any of them. Okay, no problem. Yes, remove the parafilm. Remove the parafilm. No problem. Remove the. Sit down. Bring the beaker for the disposable things. Remove the parafilm. Try keeping the plate on the bench. Okay, and closer to the. Waste, place the waste here. No, don't use this for now. You use the toothpicks. Now, you will do what? You first concentrate, focus. You will choose 5 well-grown colonies, bacterial colonies, and place them in the beaker there, okay, in the test tube there. Okay, using the toothpick. Okay. Yeah. Take the toothpick. Prepare the centrifuge tube. Come closer to him. Five to 7 colonies, it's okay. try not to puncture the gel or to remove the gel. Just okay. Spiral, like that. This is how to do spiral. Okay, continue doing that. Did the color change in the centrifuge? Check it. Did it become more cloudy, a little bit cloudy, turbid? Yeah, خلاص, close it then. Okay, good. Now, Muhammad has just demonstrated to us how to do this. Now, who wants to do the next one? Yeah. No, not Rashid. Rashid, you did your work. I want Ash-Shahwani. Ash-Shahwani did not participate so far. Yalla Shahwani.
Which tube should I use?
Which one did you use? Put it in the corner. Put it in the corner so we don't. Okay.
We use the same.
This is up to you. I so far prepared 4 different source plates for you. You can either use this same source plate or choose another one. It's okay. Yeah, okay. Open it. Choose 5 fully grown bacterial colonies. Yes, swirl until you notice a change in the color. Nassar, can you please join us here? Next time when you want to leave the lab, please tell me before. Mr. Bاسم? Okay, good. Close it, put it in the corner. Now, next student, yalla, next student. Use another source plate now. Put this source plate aside, okay? Take another one. Yalla, quickly. Remove the parafilm. Nassar, are you watching? When Nassar? Please stand here and watch your colleagues while they are moving the bacteria. Mr. Bاسم, can we have more ice? If you remove anything, please put it here. Okay, put it there in this ice bucket. Now, yalla, next student. Yalla, next student. Now, are you done with all the tubes? So this is the last one.
Yes.
Okay. Now place them in this test tube rack, in this centrifuge rack. Okay. Now they are in the ice now, right? We will transfer them now to the water bath at 42 Celsius for 45 seconds. Yalla, quickly move them here. Now from ice to warm water. Why is that? 45 seconds. Heat shock. We are doing heat shock. يلا hold it ما هو 45 seconds بس. خليها لفوق شوي. ايوه اوكي 45 seconds. measure the time. ما فيش داعي لهذا. Now, quickly return them to the ice. Bring the ice bucket. Mary, bring the ice bucket. Bring the new ice bucket to here, the smaller one. Bring it quickly. Time is critical. Now, gently remove the centrifuge tubes and place them in the ice. Yalla. Place them in the ice. We want to shock the bacteria with cooling and warming. Yalla. Place it in the ice bucket for 2 minutes. Measure the time. 2 minutes. Measure the time quickly. 2 minutes. 2 minutes. Prepare the micropipette for 250 microliter. Yalla, quickly. Now bring the sterile pipettes, the sterile tips. Bring them here. No, not this one. These are okay. Bring them. We will be using them. Now we will be using the sterile. Now the next step, you will transfer 250 microliters using the sterile tips from the liquid broth to the microcentrifuge. Okay. $250 microliter ready? Okay. When it beeps, when the time ends, start. Use the sterile ones. Aywa. Yes. What? No need. يلا ستارت. لا وليش حررته؟ خليه هون، خليه على الايس. خليه على الايس. لا طيب خليه على الايس هلا بدنا نحط لهم 250 مايكروليتر دوري بروث. يلا الترانسفر نقول لهم ايوه يلا ستارت. Prepare the other water bath with the 37. Remove the cap. يلا add. Open. يلا help him open this. No need for the flame. No need for the flame. يلا close و put here. يلا the other one. يلا open the other one. يلا لوري بروس the other one. ايوه good.
The same one?
يلا the same. Yes. Okay, good. Good, close. يلا. يلا, close it. Now put the test tubes now in the water bath at 37. Put it in water bath at 37. 10 minutes. Okay. خلاص, no problem. If it is floats, no problem. Now, step back to your laptops. Start working on the worksheet I sent to you. Start working on the worksheet I sent to you. Yes, على Teams. You will be working, it is shared files. You will be working in groups, right? Now, we have Nassar and Al-Marri working on one file. We have Al-Imadi and Al-Shahwani working on another file. And the other 3 students, they have their own also file.
طيب مين الهيفير البكتيريا ولا السوليوشن؟
Solution.
البكتيريا. البكتيريا بدون
البكتيريا بس
يعني
solution بيكون تحت البكتيريا.
السوليوشن تحت البكتيريا فوق.
This is your expectation.
دلوقتي عمادي الأوبوزيت.
Nassar says the opposite.
البكتيريا الآن
أنا أقول بكتيريا.
نفس نصار بكتيريا تحت والسوليوشن فوق.
لا مش بكتيريا هو. مش بكتيريا هو.
ليش مين أكبر؟
بكتيريا تحت. دلوقتي بكتيريا تحت. النصار البكتيريا. تمام. البكتيريا بالنسبة للسوليوشن سوليد ولا ليكويد؟ سوليد. طيب معناها سوليد يعني should be بالضبط سوليد. يعني البكتيريا بالنسبة للمي سوليد تعتبر سوليد. وبالتالي تكون البريسبتيت داون لأنها هيفي.
حطينا كذا ألوان بيصير أبيض. لو حطينا لون الرينبو بيصير أبيض.
ببين أبيض.
They will not mix together. They will get separated. If you have different dyes, different stains, according to their density. Remember, if we want to mix things together, we use the vortex, the device we used last time. Okay. If we want to precipitate things, to separate them from each other according to their density, to their weight, we use the centrifuge. Okay. The heaviest things, of course, they go down, they settle, while the liquid part, it will form a layer. Wait. خلاص. Open. Now. يعني في شوية هيك بلت موجودة في الأسفل. Check the other one. في little billet. This stands for the 5 colonies we have. يعني we have 2 layers, one clear layer and the other one is like turbid beneath one. We have 2 layers. Can you see the 2 layers?
Yes.
Can you now?
Water and bacteria.
Yes. Now we are interested in which of these 2 layers? The upper one or the lower? The lower one. That's why we need to get rid of the upper layer. The upper layer, the liquid part, is called supernatant. The one that goes down is the pellet. Okay. So we will like get rid of about 150 microliters of the supernatant of the liquid part and discard them here. Okay. And I want one of you to discard part of the liquid part of the supernatant here. I will demonstrate this to you if you hesitate. Okay. About 150. Depends on the amount of the pellet you have of the supernatant. No. Okay. Okay. Like this. I took from the supernatant this amount, this volume, then discard it here. Okay. خلاص. I return it. Okay. Now for the other ones. يلا. Don't touch it with your hand. Get the Canomicine plates from there, from the tray. هنزرعهم لا مستريكين. Yes. Okay. خلاص close it. يلا next student. خليهم هناك حبيبي. هناك anything related to the bacteria, we use this bench. Okay. Sterilize the condition because we are going to plate again right now. اوكي تركه. يلا next. هذا this one. يلا. Go sterilize the bench there. يلا يا حبيبي. خذ من الupper layer. Like this. Thank you. يلا شهواني. Yes, this. مري take هذا الستريل لا تخلي حد يفتحها. Don't open it this because these are sterile pipets. We will using now them for culturing for the last step. Okay. Now. Now, we will plate 100 microliters. We will plate 100 microliters from these into the Canomicine plates. I asked one of you to get them ready. These are the Canomicine plates. Okay. Now, we will be using the incubating loops. Now, invert them. Invert them. Okay. Now, each of you now will culture one of these plates. Okay. Now, you will take 100 microliters from one of these centrifuge tubes. Place it in the middle. Place it in the middle. In the middle of the petri dish. Then, using this sterile loop, you will spread it. Spread it in a net-like form. Okay. You would, yes. Yeah, like this, like this, like this. But then you will make a net of them. Okay. Like last time, if you remember. Okay. Good. Who wants to start? Yeah. جيب البيكر صدق عشان الديسكارد. خذ واحد من هذول التبس. التبس أنا جبتهم التبس. هاي ما هاي they are.
The yellow one or
the yellow one. خلاص. Okay. Good. Good. Discard. Discard. يلا. 100. 100. ما فيش شيء. هذا تأثير شيء. هي 100 maximum. No, it's good. It's good. They used once, place in the beaker here. Okay, good. Now, put it aside. يلا, bring the parafilm. Parafilm على البنش. شوفوا البنش parafilm. Quickly, take a piece of parafilm and wrap it around the. Take a piece of the parafilm. Using the scissor and wrap it around. Okay. يلا. يلا, stretch it and wrap it around the. No, good, good. Okay. Take another piece. Yalla, next one. Yalla, move aside. Next one. Yalla, go ahead.
Do you want to block me?
Okay. Mr.
Leave it here. Leave it here for now. We will put all of them in the incubator. Bring up the marker and label it with your name. With your initials. Put it. Leave it here. Leave it here. Don't move it. خلاص, leave it here. Yes. عادي, no problem. Yes. Eventually we will use all of them. Aside. When you want to open the packet for the integration loop, open it from this side, not from the other one. Yalla. Place your plates in the incubator and get your yalla, in the incubator there. And yes, like this. Don't invert them now, because we want them. Yalla, next one. Yeah, take this. Now wrap it around. Yeah, prepare it for him. لا, don't tilt it. Don't tilt it. Leave it like this. لا خلاص, leave it شوي. Leave it for some time to settle. Then we will move it to the. Yalla, next. No. هي حلو حلو. Before you leave the lab, don't forget to use the hygiene. Are you all done with this? انت؟
وعليه باص.
ها؟
عليه باص وبخلص. يلا.
يلا نصار. Any one of them, they are all the same. Yes. Yellow tip, yellow tip, yellow tip.
Thank you.
Mark the plate with your initials. Leave it for some time to settle on the bench. Then place it in the incubator. يلا. Are you the last one? Are you all done with this? Okay. Okay, wrap it with paraffin. Okay, label it with your initials. Now, thank you so much for being here. Thank you so much for your cooperation. Now, for the homework, don't forget to continue working the worksheet. If you have any problems, you can contact me in Teams. Thank you so much. Well done.